Right. So what kind of information, what kind of example questions that we can answer from the output tables? So we can ask simple questions. That's what majority of the people would ask. What is the change? Is there a change in the protein level? That's primarily coming from the protein expression. Like, is my protein identified by sequencing or is it identified by matching? And like how many evidence is that proteins identified with just single peptide? If it's single peptide, is it multiple sequencing events from that single peptide? These are all useful information. And also, if you are really looking into the quality of the runs, so is the machine working fine? Am I getting the best data out of the machine? So then you can also ask questions so that you can optimize the method by after looking at these kind of plots. And so you can even see, is the machine clean enough? Should I now clean the machine, or I can go on? And actually sometimes you can, let's say if you have a larger cohort study and then it's running and then it's your schedule to calibrate the machine, you can have a look and then see, do we need to calibrate or we can just postpone it? This can also be done by just looking at output tables from not even a QZ sample, but it can be also from the regular sample runs. So there is a lot of output tables. So they are all usually located in this wherever the MaxQuant run was actually performed. So in this case, these were the raw files processed, and then in that combined the text folder, you actually have all the raw files, the txt files, and then you also have this tables.txt. And if you look at this, this is giving for most of the column headers, it's giving descriptions on what exactly that is. In this case, if you look, this is actually a snapshot from evidence table, and there is this type, and that could be either whether the particular precursor is that a label, SILAC label partner, or is it just an isotopic cluster, or is it just a peak, simple peak, or if it is identified by second peptide search, or it was actually matched, so then you will see that it's a multi-match. So all this information can be identified, we can extract them from this particular table. And this is how the description look. I mean, it's for most of the headers we actually have useful descriptions in there. Right, so this is the viewer, probably Yugen will show sometime soon after lunch. And so this shows that these tables are kind of organized in different groups, let's say. And that's again coming from this, so we have to start from this. So what we actually do in our runs is we do some scans, and basically from the scans we actually get features and identifications, and then we also have some summary of the whole how the whole run was. So that's the metadata. So all the protein information, peptide information, what is the quality, like modified peptides, all of these are this identifications group. And then you have the scans, like MS scans, MS scans, MS3 scans. Then you can look whether what quality of the scans we have. This is all in this here. And you have the whole table of features, the list of all the features, and then a metadata contains further information about how the whole run went. And we can do quality control from that also. See, this is just a snapshot from viewer tab. So we can have scans, and then you have these. You can have identifications, then you have proteins, peptides, modifications, evidence, and MSMS. So the difference between MSMS and MSMS scans is this is the list of all the scans, MSMS scans. And here this is the list of all identified MSMS scan events with information on what the sequence is, what's the length, and all those score, etc. So then you have features. So there is something called all features and matched features. I think I will try to touch upon this towards the end. And then you have the metadata, where it's a summary, what's the parameters that were used to actually do the MaxQuant processing, and the whole range, this mass ranges, and we can have a lot of plots on what is the density. So is that make sense to scan from this to this, or so you can make a variety of plots. So some of the tables, actually the sum of the columns, especially the features that Nikolai was mentioning in the morning, so they will actually come up only if you say this, select this option. This is called calculate peak properties, and that if it is ticked, if it's selected, then That's an additional step in the processing, and then you get all these values in the tables. And the other option is by default, so most of the people do not need these tables, and then it's mainly protein groups, peptides, and associated information. So these tables are primarily not used by many people, so by default it's off, and if you want, you have to select them for analysis. Right, I think maybe I stop after this slide. So this is the protein groups, that's the biggest at the highest level, and then that's consisting of many different peptides, and the peptides can be either unmodified or modified, and they come from multiple evidence, and that's actually coming from a lot of identified MSMS spectra, right? So I try to show this as an example. So now it should be. Yeah, you see the mouse. So what I have done here is actually put together all the identifications tables in one Excel sheet. So that's the protein groups, peptides, modification specific, and all the further tabs. So if we look at the protein groups, I'm just randomly filtering for 2 proteins, and then these are the 2 proteins, and then we generally have information on what's the number of peptides, unique peptides, razor peptides, and then if we have multiple experiments, that's again just repeated for each experiment. Then what's the size and what's the identification type? In this case, actually it's all identified by MSMS. And the sequence coverage in the different experiments. Then comes the intensity, then comes the LFQ intensity, and the number of MSMS counts for each protein. And then that's this multi-numerical table that's actually linking this particular protein groups to the different. So this would be the ID. So for example, this is the ID of the protein in this table, and that can be linked to these IDs in the peptides table. It's kind of a relational database if you can construct with all these tables. Right, so if we look into what's the peptides that we get from these 2 proteins, they look like this. And these are all the peptides that we get, and then this is some N-terminal cleavage window and C-terminal cleavage window. So sometimes it's useful to see where the particular, if you want to see some motif which is present in the protein that you can see. And sometimes it's empty because the N-terminal cleavage window is empty because this peptide comes from the very beginning of the protein, so there is no N-terminal cleavage window, right? And also this is like, this can produce a sequence tag, like a motif tag for what's the protease used and what kind of cleavage that actually is present in the sample. Like in this case, the amino acid before the particular peptide in the protein is lysine. So it tells us that it's after lysine this was cleaved. That's the first amino acid, second amino acid, similarly the last amino acid and second last amino acid. So this is a triptych sample because you have either lysine before the peptide or arginine. So this can be used when you have an unknown enzyme or you're testing new enzyme and you want to see whether it's specific or it leads to a lot of unspecific cleavages, then this particular signature can be used to see in a global level. Then there was this just amino acid counts and length, what's the miscleavage, mass, and all the associated information in this. We will use some of them to make some plots and see how this can be used. And again, you have the intensity information. Yeah. Good. Then, so we can have actually some peptides, and then that's unique in sequence, but maybe it's not unique in terms of mass because this was identified as oxidized, it was identified as unmodified. For example, this particular peptide, it's exactly the same in terms of sequence, but it was both identified as oxidized and unmodified version. So then this is also listed. So that is this modification specific peptides. And then for all the entries you have, for all the peptides you will have, if there are multiple versions of the same sequence, you will have information in this. But now let's imagine oxidation is one of the major modification important in cancer. because this is a regular sample. So, and then let's say if we are looking at that particular modification, so this is the modification sites table. The sites table is at the level of sites, like looking at the position of the modification. Like for example, in this case, there is one, this is the protein, and in that protein there is this modification with a localization probability of 1, which means right. So in this case, the peptide was identified with methionine oxidation. There is only one methionine, and therefore it's pretty sure it's here, and therefore the localization probability is 1. And yeah, it's a bad example to show something less than 1 because most of the methionines are modified, right? And again, it's the same kind of continuity about identification type, intensity. I think intensity 1, 2, and 3 probably Kirti will cover what they are. And yeah, and if we take one particular site information, then we can have a lot of evidence. So this is actually exactly the same sequence, and then let's say we look for only the unmodified, and let's look at one particular sequence. Then you have many evidence, and that's coming from sometimes the same peptide is sequenced in different chart states. These kind of information you can get, and sometimes actually this is in some raw files this is sequenced, and in some raw files this was actually matched into. And just a quick question, so if I am analyzing single raw file here, what would be missing in this thing? Like what happens to the multi-match? If it is only one raw file in this MaxQuant analysis, will we have any multi-match? Yes, no, maybe. Okay, so the answer is no, because it's only one file, so there is no matching between the runs, so you will never see this particular evidence in that particular table then. Right, so in this, so then the evidence table is becoming closer to the mass spec properties, like spectral properties, because you start getting the mass error, uncalibrated mass error, and what like retention time, length, retention time calibration, number of data points. This means essentially, roughly speaking, this is the number of times the mass spec actually saw that peptide eluting in the chromatogram. Right, so that's a lot of information here. And then the evidence comes to the MS scans that are actually identified. This is roughly the same, but then you start to get information like that's a CID fragmentation, that was an ion trap as the mass analyzer, and what's the scan event number. All these information come in this particular table. All right. Do you have any questions? Yes. I was hoping nobody saw that. No, there is no. Yeah.
So if I want to look at the modified peptide LFQ intensity, how do I do that?
At the moment, there is no LFQ intensity reported for modified peptides. So for example, even for the phospho, you just use the intensity as such. You don't have any LFQ intensity.
But then do I use the intensity for, let's say, like for the entire protein group, like for the unmodified peptides as well? Because I cannot, so my question is, when a user comes and wants to look at the phosphoprotein in the global proteome setting, not enriched phosphoproteome, just want to look at the modification as a first pass, I have the protein, like in the protein groups, and then I go to the phosphosite to only see intensity. So then when I do the global proteome search, not phospho, just all the proteins, should I just take intensity or use LFQ in one case and intensity in another
I think for
case?
the proteome, you would use the LFQ if you have the LFQ, right?
Like sometimes, let's say the protein increased and the phosphoprotein also increased, like the phosphopeptide also increased. But if one is LFQ and one is intensity, and if I'm normalizing that, I don't feel comfortable doing that. Like,
Well, there is no systematic, I mean, there is no easy ready-made workflow here, but you can simply check, okay, so now this is my, it's also a little difficult because you cannot really compare the different peptides intensities, but approximately they are in one, within 2 orders of magnitude if they fly bad or worse. And then let's say the phosphopeptide has certain intensity, and then you can see is that counterpart unmodified peptide of this phosphopeptide, is that going down or is it in that average cloud of the other peptide intensity? That tells whether the phospho is going up on top of the protein quantification.
okay.
If I could just go on that, what is the basic question? Are you worried about is the change at modification site level or is it at protein level? Is that your question? Because it depends on the question. Where would you go and look for what kind of data then?
Okay, I can talk to you later.
Yeah, sure.
Yes, question here.
I have a question about what exactly the intensity for the precursor is. I've always assumed that this is the integrated precursor ion area, but in the table's description it says intensity. It doesn't confirm that, so I just wanted to be sure what that column is.
I think it's the peak intensity, right? It's the peak intensity of that particular precursor in that particular scan. Huh? No.
In that
Yeah, peak intensity.
peak intensity, is there somewhere integrated precursor ion area? So that would be maybe for Siloc?
I think there must be a setting somewhere where you can switch between
Yes.
how the intensities are calculated. So it's either value at the peak or it's integrated area. And I think there are two more options. Like first you model it with some fit and then it takes the maximum of this. I can show you the options later.
This question there.
And my second question is about the mass error. When that makes a lot of sense and is very useful for label free searches, but when there is TMT labeling, and the mass error seems to include the mass of the TMT tag as well, and then it's on a ridiculously high scale. Is there a way to get the actual M over Z error that doesn't include the TMT label? Okay.
So my question is, when you have done a large comparison and you've identified a handful, say 5, 6, 10 proteins that you're interested in, and you want to go back and verify with one other evidence, I guess. So you have your proteins. Which table do you go back and say, okay, I've got 3 peptides here, 6 peptides there? How are you, what are you going back to convince yourself that your changes are correct?
So usually, I would say the short answer is I would say there is, let's say it's not a very lowest abundant protein that you're looking at, and you have a drastic change. You will not verify. You go back and verify, but essentially the information is coming from the tables in a backward manner. So if you want to see how exactly you get this, so you can, for example, in this particular protein, let's say, I think one of them should change because CTC48 sounds like it will change. Unfortunately not. Okay, so let's just take this protein and then now you want to verify if everything is correct, right? So to verify if the LFQ calculations is correct is difficult because we cannot go back and check how the LFQ is calculated. But if you already see that, okay, so there are 16 peptides, which I would also go against it, but for now I would say 1624 peptides are there. So whatever quantification we get out of this is for sure correct. But this is also speaking against a protein which is changing a lot, but it has only one peptide because of just the protease choice. Then it's also difficult to decide on that. So I don't know. I'm not even answering your question, I guess.
Well, I guess that's why I'm asked, because it seems like a lot of the times you're digging for those less abundant proteins that are important, and you find something with maybe 2 or 3 peptides identified for it. And I'm like, is there a way to visually
Yes.
go back to one of your other
Yeah.
tables and get
Now
a
I
quick
get it.
look somehow?
Yeah, I get that question. So let's say if we have a low abundant protein, and then, for example, let's say we have one peptide identified, so we can actually go to, for example, even in the evidence table and see, do I have, let's imagine that's the only peptide that you get, and that's the only evidence you get from that particular protein, and then you can look at this n points or something like this. Can somebody of data points or n points? I cannot do this with 100 people watching, so
Sorry.
yeah, here. Number of data points. So in this case, it's like 124. That means at least in 120, number of scans is 26. But then it had this particular peptide had C12, C13, this isotopic cluster. So then at least 124 times we can actually see that peptide. So which means that gives a confidence that it's not just one time looking at a scan. So you have information from 124 measurements. Right, that's way better than just one Western blot.
Okay.
Is that
Yep, but how about, say I have an N of 5 for each group,
yes?
and I want to make sure that it's in 3 of 5, maybe at least for one group,
Yes.
and see a change somehow.
Yeah, you can. Yeah, that's also a perfectly valid thing to do. But if you're really looking at, is that protein first of all correct? Then this is what, if you're really suspicious, you can even look at this. You can look at the mass error, and so the mass error is what? 0.2. Not so great, not so bad. So
Is there some kind of graph that you would actually say somebody wanted these evidence? Say one of my customers wanted evidence for their half a dozen proteins. Is there a way you would graph some of this out?
in such situations, I usually don't do the graph. I just simply send the evidence text as a supplementary table if the reviewer would ask.
Okay,
That's sufficient. Yeah.
thank you.
Right. I think yeah. Okay.
So I had a question about kind of cross-referencing these different tables. If you had a peptide that is in your peptides table, and then you also have it in your modification specific peptides, are the intensity that is associated with it in the peptides table going to just be the sum of its appearance in other tables where it might appear multiple times?
This I would say I don't know. It's a sum. Yeah.
Okay, thank you.
So in the meantime, I keep the next thing open. There was a question somewhere. Okay. Right. So I quickly want to show that this is a very quick snapshot on the modification specific peptides table and the site table. I just want to show an example because Kirti will anyways go into the details of this. This is a study on some cell cycle specific project, and then here, this is a protein CDC16, and there are like about 1, 2, 3, 4, 5 possible positions for one particular phosphorylation, but then since they are so close to each other, if you look at the sequence, yeah, you cannot see this properly. So there are 5 positions, and then since none of them can be decided for sure, they all get a localization probability which is much less than 0.5. So in such a situation, you don't get any intensity value reported into in the table if you are looking into this. But if you actually are not keen on looking at this at specific site level, but at the peptide level, modified peptide level, because then now this actually there is intensity reported for this, and that can be used. So you're instead of treating this at the site level, you're actually treating this at the peptide level. And so that you're not, so, and this in this case actually the trend was perfectly how the user wanted to see, and so very happy to use that particular modification sites, modification specific entry, because otherwise you would simply discard this because there is no intensity reported in the site specific table. Right. Going back to the slides. Right, so this is what we saw, like it's, that's the same thing, so it's protein identification and quantification, that's in the protein groups table, and if it is label free, you get this LFQ intensities, if it is SILAC, you get the ratios, and if it is TMT, you get the reported intensities, and then, yeah, then you can see whether the protein is identified by matching or sequencing, and yeah, and then this is the sites table that we saw. Okay, so sometimes from the peptides table, a lot of information can be obtained. So for example, what's the score and cleavage window, this we saw, and then protein centric. So we can actually see what's the start and end position of the peptide within a protein. I will show an example why we need this. So we can make just histogram plots of the length. This is all coming from the peptides table. Like missed cleavage rate, just plot them and see how frequent is this. And then in some cases, just to check if you have missed cleavages, probably not use up maximum missed cleavages of 2, but maybe like 5, so that you actually open up the window to see it if you have more missed cleavages. That would be nice. Yeah, this we saw. Okay, so in the peptides table, for each and every peptide, you also have the start and the end position that is reported. And this is pretty useful because let's say if you have a protein and then you want to see from which part of the protein to which part is actually identified, so then you can simply plot the X start versus the end position. And so here's the start and the end, then you see that you almost, for this particular protein, which is about 900 and some amino acids length, so you see that it's completely full length. But we can also do the different thing that we take the start position and then plot that against the peptide's intensity. So now here, I mean, we have to be a little careful that each and every peptide can have its own intensity. But if you look at the plot, we see that it looks like predominantly the C terminus of the protein is still remaining and the N terminus is kind of gone. It's just a leftover. So by looking at this, we know that it's not a full length protein in the sample. This is actually the C terminal fragment of the particular protein. And maybe some of you are doing limited proteolysis and to see what is actually left in the sample after limited proteolysis. So then in this particular case, these are different samples with different treatments. That's the untreated, so you can see the full length. So basically this is the start and end position that is used here for heat map. And the red is higher intensity. So now we see in that particular condition with certain concentration of the protease, and then upon digestion, so we still have only this part left. So probably this is not exposed to the solvents or something like that. These kind of information you can get from the peptides table. And again, so like if you're looking at fractionation efficiency, if you do a peptide level fractionation, and then you want to see are we getting unique peptides in each fraction? So then we can simply use the intensity as in each fraction of the peptides and then cluster them, just do a heat map, and then this shows what is the fraction overlap. I mean, here just a small tip. So if it is the fractions are used and then you need the intensity, then each fraction should be run as an independent experiment. But probably this we will show later in the MaxQuant setup. Right. Any questions here? No. Right, so evidence we saw, it could be multiple sequencing events and different chart states, unmodified and modified. And then you also have these information. So you start to get this PIF, this what is the purity of the precursor that you're fragmenting, and what is the base peak fraction. So all these information is to actually can be used to actually see should we change the mass spec method, should we change the intensity threshold of the MS2, should we increase the number of MSMS and all those things. Right. Yeah, so we can actually take the mass error that is reported in the evidence table and then plot them according to the retention time to see if it is all systematically like this or it's like sometime deviating. So all kind of investigation can be done from these tables. And if we have two runs, we can check if the retention is if the LC is reproducible by just looking at the retention time of the run 1 and retention time of the run 2. And most importantly, I think in order to check the reproducibility, you should actually take the uncalibrated retention time and not the calibrated retention time, because when you take the calibrated retention time, probably everything is within 2 minutes. So that's another application. So then we have scans. So some of the scan examples. This is just a peptide actually eluting in this particular time. And you can actually maybe select this for fragmentation at different points. So this could be selected at the top. Then that is actually the apex fraction, approximately is 1. Yeah, because it's at the top. And here probably it's like 80%. So and then here it's at the bottom. So then this information is present in the in the evidence table. It's also present in the MSMS scans table, because that can be used to see at what apex limit we start not identifying any spectra. So these kind of information we can get from these tables. Right. So again, if you have a very complex spectrum, and then are you digging too deep? Let's say, is that worth? So for example, this is the base peak, and that's the base peak fraction 1, because that is the base peak, and then this has a base peak fraction of 0.5, and that's pretty low. And then we can see to what level, so should we do top 10, top 5, and we can see, is that really worth doing these MS2, whether are we identifying that spectra? So if not, then we don't have to even, we can change the method to slightly avoid these MS2 spectrum, which are anyways not identified. And that is the purity of the precursor isolation, which is kind of crucial. So if we have two different peptides eluting at the same time, and this is the isolation window, then so we really have to know what is the PIF, the purity of the peptide, and that is probably crucial when you are doing TMT labeling and TMT quantification. Maybe you want to filter out all the precursors which are below a certain purity level, so that you don't have ratio suppression. Right, so this is actually from the MS scans table. I will try to explain this. So if a peptide is actually eluting in the retention time scale, and each circle here is actually one full MS scan. So this is so we actually have in this case 1, 2, 3, 4, 5, 6 snapshot of this particular peptide, and probably this is too less. And each full scan, and then the time between the full scan, you do the MSMS scan. So this is actually the cycle time. The cycle time is between 2 successive MS scans. So we can actually see if we are doing top 10 MSMS events, what is the cycle time? If we are doing, okay, don't have anything else. Okay. So this is, if it is doing top 8, this is the cycle time. So this gives an idea of if I do so many MSMS scans, what is the cycle time of the run. And why is that it's useful? Because we can see that also with the different top ends, we actually also see the number of data points that we get for certain peak that can also change. So you have to see that you are fast enough, the cycle time should be fast enough that you are not losing too much information. So you should have enough snapshot on the peak so that it's good for the quantification. And also this is useful, let's say you were running 60-minute gradients and now you want to go to 20-minute gradients. So then we have to really rethink, are we still going to do like 1 second cycle time? Maybe we will get 3 data points instead of 6 data points or 20 data points. So all these information can be used to decide, is that method correct, or is that method probably needs a change in the gradient length or the number of MSMS that we do. Yeah. Right, so this is actually an example where this is the same cycle time, and actually we see that at the beginning it's pretty fast. The cycles are it takes less than 300 milliseconds, and then at the top it's taking like 1 second. And then if we also see the number of MSMS scans happening here, this is also much low, which therefore the cycle time is low. And the reason why the number of MSMS scans are low is probably because the actual population of peptides that are eluting at that time is less, right? Which kind of indicates that probably we have a suboptimal gradient where there is nothing in the beginning and then suddenly it starts to rise up. So which means with the first 25 minutes is actually a waste of time in this particular gradient. So we actually have to change the gradient so that we can make use of the full machine from right from the beginning till the end, right? So that's another example. I think, and then there are like features, and features is basically a list of master charge, intensity, the whole features present in the run that can be used to plot these kind of histograms. And so here it says this is the all features. The green ones are the ones which we are actually identifying, and the red ones are the ones where we actually try to identify them by fragmenting them, and the gray ones are not even touched for fragmentation. So this also gives an idea of is the machine fast enough for this particular sample, and other information from this. Right, I think yeah, so then there is the summary table that gives an idea of what is the number of MSMS scans, what is the identification rate in the sample. So this is actually in the summary table. So for each run, what is the number of MSMS scans and what is the number of MSMS scans submitted, and if they are all they have Silac pairs or isotopic clusters, or they are just single peaks, and what is the identification rate, and this information is available. And actually from this, if you see, if you see the if the ratio of the cell to the peak is too high, or like if you have more than 1/5 of the spectra here, how do I say this? If the number of spectra in the peaks column is more than 1/5 of the number of spectra in the cell column, that tells that in lot of peaks we are not looking at the isotopic cluster, we are just looking at the C12 peak maybe, which means maybe that many of the peaks are too low, or maybe there is something wrong in the analyzer, in the way we fill the analyzer, maybe something is wrong. So that's a good indication, and see what's actually happening in the machine. And then the identification rate can also be a good indication of how well the machine is performing, right? Yeah, so this is all the kinds of table that you can get from all the processing. And if you have any more questions, I'm happy to take. That's the end of the presentation. Thank you.